Okay, thank you. So I'll give a brief introduction to why I look at cyanophages, or used to look at cyanophages. So this here is simply an image taken down a microscope of seawater. And all these big blobs here, these are bacteria, and these small specks, these are viruses. And marine viruses, and viruses in general, are the most abundant biological entities on the planet. It's estimated to be around about 10 to the 31. So this is just 1 ml of seawater, no concentration. There's probably about 10 to the 7 viruses and bacteriophages in this seawater sample. And this is what I'm particularly interested in. And why am I interested in? And it's because of the ocean dominates across the globe. In these big regions here, these big dark patches, this is where you find cyanobacteria. So cyanobacteria are phototrophic, so they produce oxygen and fixed carbon dioxide, and they're extremely important in biogeochemical cycling. In these open ocean regions, although there's a low overall net primary productivity, due to the vastness of them, they account for huge amounts in a global scale, more so than the tropical rainforests. And it's viruses that infect cyanobacteria that I'm particularly interested in, and they are known as cyanophages. So in these big dark regions that I showed you, the two main genera of cyanobacteria are Synechococcus and Prochlorococcus, and in some regions these can account to 80% of primary productivity. And at any one point, up to 40% of these cells may be infected by a cyanophage. So this is the background of why we study these cyanophages. So I've mainly worked on a model system using this particular phage and cyanobacteria, and the cyanobacteria is known as Synechococcus Woodsolve 7803. So it was isolated in 1978, and it was the third strain. It has a nice vivid red colour when it's grown. We have a complete genome sequence which has been annotated and around for a long time now, and we use this as our lab strain. And the model system for the phage is S-PM2. So it infects Synechococcus. It was isolated from Plymouth marine waters. It's a myovirus, and it was the second one that was isolated from a particular sample. So it was isolated in the early 1990s by Willie Wilson, who used to work at the University of Warwick. And at the turn of the century, somebody else in Nick Mann's group found that it had a shared genetic module of structural genes with that of T4. So T4 is an enteric phage affecting E. coli. So they look fairly similar, and this is largely due to the fact they share this conserved module with structural genes. What's different about S-PM2 is, and what I'll come to talk about, is it contains photosynthetic genes, and I'll explain what these do. So we have the complete genome sequence of S-PM2, which we completed in 2005. Some of the differences between T4 and S-PM2 is that S-PM2 is a lot bigger, has a far bigger genome. It also replicates far slower than T4, which is less than 30 minutes. We're looking 5 over 10 hours. For S-PM2, or 9 to 10 hours. It has a large number of auxiliary metabolic genes. It has no toxin-antitoxin system, whereas T4-like phages do. It has no CRISPR system in Synechococcus, where you get CRISPR systems in T4. So these environmental phages are quite different. So I'm mainly going to talk about these photosynthetic genes and psbA and psbD, and explain why light is important to cyanophages first. First of all, if you don't have light, S-PM2 cannot absorb to Synechococcus, so it can't infect it. And what's even more interesting, this is actually a wave dependent, so different wavelengths of light allow absorption rates change. But the main focus is going to be on psbA and psbD, and these encode for the proteins D1 and D2, and these are found in the Photosystem II, in the core of Photosystem II. One of the problems, one of the things common to all photosynthetic organisms is that these proteins D1 and D2 get degraded during photosynthesis, and they have to be constantly turned over. Otherwise you get photoinhibition, and if photoinhibition occurs, you get less energy production. So when we discovered these psbA genes, the theory was that the psbA gene is expressed by the phage to maintain its host to allow replication to continue or possibly give an advantage under a certain condition. But what this means is that if you do calculations on the number of phages present and number of cells infected at one time, between 5 and 32% of primary production may be driven from a single phage gene. This is a video that sadly isn't working, but basically what happens is in the center of Photosystem II, which you have here, you have this psbA protein or D1, and this gets degraded by light as a result of photosynthesis. So it gets excised, it comes out, and it gets replaced by another copy of psbA or D1. And we think this copy is often the phage copy, and this is what this work's going to focus on. So this is the psbA region in S-PM2. So we have our psbA gene, and within that gene we have an intron, which is quite unusual because most bacteria and phages don't tend to have introns. We then have homing endonuclease, a hypothetical protein, and a second photosynthetic gene. And what I've also found is that we have CFR1, and CFR1 is a non-coding RNA that runs from the homing endonuclease back into psbA. And at the time it was unknown how far back it ran into psbA or whether or not it had any function. So as I said, psbA is thought to have a fitness advantage under certain conditions, and we think the most likely one is high-light conditions. So we know we have this non-coding RNA. What we didn't know is whether or not introns are widespread in psbA genes. So psbA is used as a marker for cyanophages now, and people have sequenced literally thousands of psbA genes, but they never seem to find another intron. So we didn't really know why this was the case, and whether this intron is capable of cell splicing, and whether the intron has a function. So I took the GOS dataset, which was produced by Craig Venter when he decided to sequence the ocean. So this is a massive database of sequences and contigs that are found in the ocean, and I started looking through these to try and find genes to see if I could find other introns and psbA genes from phages, and you can find a fair few of them. And this is just some of the contexts that you find them in. So you can find, this is S-PM2 in the arrangement, so if it's the same colour, it's the same gene, or we think it's the same gene. So you can find a psbA with an intron, and you can also find this homing endonuclease next to it. Sometimes you don't get an intron within the gene, psbA gene. Sometimes you get different arrangements of them, but in the environment you can find introns within psbA genes. So what is the interest in this psbA gene? So I did mention that it was a homing endonuclease. So at the same time I was doing this work, a group came up with how they think that the mobile group 1 introns are formed. So this is their overarching view of how they think they're formed in bacteriophage. So you have an ancestral gene arrangement, and within this there's a target. So this homing endonuclease can target this specific site within a gene and insert itself next to it. So all a homing endonuclease is a genetic parasite. It just tries to get from one piece of DNA to another. It doesn't have any function that anyone knows of. It just tries to move about. This same site is often targeted by an intron. So the intron can insert in the same site. So what they think happens is that you have one population that has the gene and an intron gets inserted at some point, and you have a separate population where you get the homing endonuclease that is targeting the same site that the intron inserts into. And then during mixed infections, you'll end up with an intron in the gene, and then next to it you'll have a homing endonuclease. So the advantage of this to the system is this: the intron prevents the homing endonuclease nicking or cutting its own DNA. So it's a protection measure, but it does still allow it to propagate into other hosts when you have a mixed infection. And then to form this true mobile group 1 intron, what you can have occurring is a homing endonuclease inserting into the intron, so you can splice and also target new genes in one step. And if you look at T4 and relatives, that you can find all of these steps. So they think this is quite a good system of explains how they form. So if we take the data that we have for psbA and look at this, we can find an arrangement where we have psbA on its own. We can also find psbA with an intron inserted it. We can find psbA and a homing endonuclease but no intron. and we can find the psbA homing endonuclease in the same system, which is S-PM2. Well, we tend to find an antisense RNA that links these two systems. What we never find is psbA, an intron, and a homing endonuclease inserted within it. So is there a reason for this? Does this antisense RNA prevent the formation of a true mobile group 1 intron? Well, if this antisense RNA had a function, then this may well be the case. Just to add to the slight confusion of this, if we look in the metagenomic data sets, we can find a psbA gene, we can find a homing endonuclease, we can find an intron, but we find a different homing endonuclease family gene within the intron. And we can also predict this antisense RNA. So this gave some credence to, we think this antisense RNA may have a function. So one of the other questions was, is the intron in psbA spliced? So if it's not spliced, then you're not going to get a functional D1, and if you don't get that, then the whole theory of D1 providing an advantage to the phage is going to fall apart. So using a fairly simple technique, extracting RNA, using reverse transcriptase on these samples, you can identify both the spliced and the unspliced version of the psbA transcript during infection. So this is spliced at 1, 3 and 9 hours during the infection cycle. So we know that it is capable of splicing. But what we don't know is the intron spliced at different rates in different conditions. And does the CFR1 run back through this intron or interact with it in any way? And does the psbA gene actually provide this fitness advantage that we think under different light conditions? So this is an experiment of Synechococcus infected with S-PM2 under high and low light conditions, which we've defined as 20 microeinsteins, which is what we normally grow the cyanobacteria at 300 microeinsteins. So this is just a level of light. If we look at the S-PM2 infected at 6 hours, you can see it's slightly paler than the control with no S-PM2 added. And at 9 hours, it's pretty much clear, which is indicative that the cells have lysed and the phage is replicated. If we compare this to the lower light condition, then you can't really see any difference. So we've got phage in the S-PM2 sample, but it doesn't look anywhere near as lysed. This is some preliminary data using qPCR to determine the number of copies of DNA that are present, or S-PM2 particles that are present, and under this highlight condition we can clearly see we get a massive increase at about 7 hours compared to this low light condition. So we think under highlight conditions that the phage is replicating faster or producing more phage, and when we get the one-step experiments we'll be able to identify exactly what this is. So does the psbA gene that's encoded in the phage do anything different? So one way to measure this is to look at FV over FM. So this is a measure of the photosynthetic capability of the cell. So it's the variable fluorescence divided by the maximal fluorescence, and it's a good indication of how healthy a photosynthetic organism is. So we have high light and low light infected cultures, and what we're able with and without phage, and what we're able to show is that in the high light conditions, the phage, if phage is present, it maintains the same, or it maintains a higher photosynthetic capability compared to the low light conditions. So it's suggesting that the phage is providing some mechanism to have a increased or maintain the photosynthetic ability of the cell. And if we look at the ETR, which is the electron transfer rate through Photosystem II, in the plus phage condition under a high light, you get more electrons being shunted through the electron transport system. So we think this psbA gene is responsible for an increased or maintained photosynthetic ability of the host. If we look at the expression of psbA, we can see that under a high light condition, the psbA increases during infection the same way that low light does, but there's a bigger increase in the expression of psbA. If we look at the unspliced psbA, so because there's an intron, there's always going to be pre-mRNA present. Again, in a high light condition, we get an increase compared to the low light in the amount of unspliced psbA. So this raises the question, how is this maintained? And is there any particular reason for it? So if I then go back and look at the antisense RNA, so CFR1, we can see that again in a high light condition you get more of this CFR1 compared to the low light condition. And if we look at the ratio of unspliced to spliced psbA transcripts, under the green highlight condition, there is a greater pool of unspliced psbA transcripts. So if you want to respond to an environmental stress of high light where you need to turn over your D1 more, by having this pool, it would make sense because you can quickly turn this into a protein rather than have to. Whereas in the low light condition, there's a far lower, so you initially get an increase, but then it quickly drops off. So how is this pool of psbA transcripts or unspliced psbA transcripts maintained? Well, we think the answer may be the antisense RNA CFR1. So this RNA, we now know where the end is. So it starts in the homing endonuclease and it will run back all the way through the intron. It's quite long and 3/4 of the way through psbA. By mixing the psbA transcript and artificially produced antisense CFR1 transcripts, we can show these bonds. So we made, Rich Puxty, a talented PhD student, made a large number of transcripts of different length of this antisense RNA. And if we have a very short one, what we can see is here, this is a control. So this is a spliced psbA transcript. There's an unspliced psbA transcript. And this is the intron that is of interest. If we have a short one, we don't stop the intron splicing. As you increase the length, when you get a full length antisense transcript, it prevents the splicing of the intron. So we think that this antisense RNA may bind to the psbA transcript to prevent the splicing. So this is a mechanism to maintain this pool of unspliced transcripts to respond to light. So we now know that psbA expression increases under high light conditions, that we have a pool of unspliced psbA maintained under high light conditions, and that the CFR1 increases under these conditions, and it may be the binding of CFR1 to psbA that offers a means of regulating this splicing to maintain the pool. And we also think this adds to why CFR1 prevents the formation of a true mobile self-1, true mobile Greek 1 intron. So I'd like to thank the people that I've worked with over the years. So Nick Mann, who originally started this work, and then Dave Scanlon, whose group I moved to, and Rich Puxty, who's a PhD student who's been working on this project recently.